Uh, am I audible?
Yes, sir.
Priya, am I audible? Priya Chaudhary?
Yes, sir. Yes, sir.
So I think our first semester has joined, I think. And Devojit, am I audible? Devojit, Devojyoti.
Yes, sir.
Can I proceed? And I think Joydeep, the streaming has been started.
Yes, sir.
Okay, so I think this is our tutorial 2 with me, assistantship. Shormishta is with me today, and we are going to start our understanding microscopic imaging for cancer classification and demonstration of the mainly ground truth creation of cytological images and histopathological images. So I think Devojit, am I audible? Devojit, Majidul. So we are going to start. Ronit, Ronit, am I audible?
Yes, sir.
So we are going to start. I think all the participants are not joined, I think. Okay, so we can proceed with that is the today's outline of this tutorial. Microscopic imaging already have done by the Professor Nath, what kind of thing. And I think our objective is how to link the imaging, cell imaging, with the artificial intelligence and machine learning. It is our main motto. So I think anyone tell me what is that is the cytological image, what you have learned from the session 1 and 2. Devojit, Ronit, can you tell me? Okay, so then these two things we have already learned a lot from Madam: microscopic imaging, histopathological image, and papsimer image. So we have our own research today we are going to present. So we have a lot of work in cervical cancer papsimer images with a standard data set, that is the NORUP data set, and our own developed data set with Agartala Government Medical College and Tripura University dataset. And Shormishta will assist me, our research scholar, and she will also show you some hands-on how to create the annotation of the cellular data, basically. So I think data acquisition, ground truth generation with a GIMP uh, tool. So this is the total layout. So as you know, we are not going to discuss anything about the cancer. You know a lot from these two sessions. So we are going to basically what we see in our naked eye, which is not possible by the computer. Whenever you will not code anything, the simply we are going to present here. One is PEP, SIMR cytological imaging, and another is histopathological imaging. So here is a one cell, and histopathological imaging, there is a lot of cells. Cell plus cell plus cell plus is called with other accessories or the peripheral is called histopathological images. And in Pap-SIMR images, only single cell. So we have work done already a lot in histopathological images and Pap-SIMR images also. So our observation on the two broad category. So in the thing is that how can we proceed with machine learning with this kind of data set? So one of my question as an interactive session, anyone tell me what is segmentation or the classification? Devojit, can you tell me what is classification? Devojit, anyone tell me what is classification, what is detection, and what is segmentation? So okay, so that is the main thing, going to why machine learning is most important, to learn the complex features. So what kind of feature here in the cytological images and the histopathological images? So machine learning models can achieve a higher diagnostic accuracy compared to manual observation. So what kind of manual observation? See, it is one of the, I think, benchmark data set, that is the Breakage data set. To see this kind of data set, we can't know that is the which one is the cell part, which one is the number of cell part, and which one is the lump, and which one is the single cell. So this is a standardized, and they have created their own annotated data or the ground truth data with the normal data. So today is our main motto to create the ground truth data. Now you see here, as you know the previous lecture, regular shaped means the normal data, that is the size of the nucleus and the size of the cytoplasm is given. But in the irregular shaped, it has been seen in the cytoplasm nucleus part is increased. So it is the part, one of the part of histopathological imaging that we have present here. Now this is the Pap-SIMR images. You know the famous scientist, that is Papa-Nicolau SIMR. From his name, this is called PEP, and this is called SIMR. SIMR is the cells imaging, is a cytological imaging. So here it is. If you see, use my cursor, this part is called the nucleus part, and the surrounding part is called cytoplasm part. Same as this was that is the regular shaped nucleus, and the surrounding is called the cytoplasm. But compared to the normal data, if you see my cursor, this is the nucleus is spreaded, overspreaded. Okay, and this is the shape of the precancerous or the cancerous condition. That is 90% cytoplasm is covered by the nucleus. The size of the nucleus is increased here. This kind of thing is called cancerous or precancerous cell. So in a naked eye, you can see in a computer vision which one is which cellular part is cancerous and which one is normal cell, normal epithelial cell. So now, can we ground to them? So now I will hand over to Shormishta. I will hear also. So Shormishta, you can proceed to uh, how to create the ground truth or the annotation of this kind of data. Shormishta,
Yes, sir.
okay. Now you can proceed. So Shormishta will tell you that and some hands-on experience how to create the annotation. So you can proceed, Shormishta.
Everyone,
Okay,
myself,
sir.
Shormishta Chakraborty, research scholar of Department of Computer Science and Engineering Department, Tripura University. Today, I will assist Dr. Minalkanti Bhumik sir in this tutorial session. So, among various types of cancers, cervical cancer is the most common disease among women. Cervical cancer is a cancer arising from the cervix, which is mainly caused by infection of several types of human papilloma virus. It is a malignant tumor that starts in the cells of the cervix. Malignant means that it can spread to other parts of the body. Cervical cancer is curable if it is detected in early stage and treated appropriately. Cervical screening using pap smear images is one of the most effective ways of detecting and diagnosing the disease even at an early precancerous stage. We are tasked to detect this disease at the precancerous stage. Cytological based screening methods like pap test have been the major approach in a clinical test. But these screening methods must be performed regularly. The main purpose of the pap smear is to detect abnormal cells which may develop into cancer if left it unchecked. Pap test is a manual procedure where pathologists visually examine the microscopic slides and identify any abnormality in cell structure. The visual interpretation of microscopic images is usually labor intensive, time consuming, and subjective. In the visual analysis of cells in pap smear, the correct characterization of microscopic slides and drawing of any conclusion thereof depend mostly on the general appearance of the nucleus of the cells or the sizes of the cell. From the pathological point of view, the nucleus of infected cell may be enlarged with a possibility of increased DNA content or irregular chromatin density and irregularity in shape. So here you can see the features of normal and abnormal cell in the if the abnormal cell is irregular or the shapes may vary and the size of the abnormal cells may be enlarged, whereas the size of the normal cell is relatively consistent among the cells. And also the cytoplasm the irregular boundaries with dense cytoplasm. And the nucleus will be also be enlarged with respect to the with respect to the normal cells. Here, in the first cell, that is the figure 6A, this is the normal cell, and figure B is the abnormal cell, where in both cells, the nucleus area is marked by the red color and the cytoplasm region is marked by the green color. So you can find that in the first one, that is the normal cell, the area of the nucleus is very small and it is also centrally aligned. Whereas in the case of abnormal cell, the nucleus area is enlarged and it is like a oval structure. And in the first cell also, the cytoplasm area is clear. Okay, and here in the abnormal cell, the cytoplasm area is dense and the boundaries are also irregular. Fast computer-aided diagnosis of cervical cancer necessitates to create a cervical cancer database. However, till now, there are very few databases of cervical cancer has been reported all over the world with less number of samples. One of the most widely used benchmark dataset is Neurope dataset. So it was actually created to support the development and evolution of the automated system for cervical cell segmentation and classification. It helps in early detection and classification of normal and abnormal cervical cells. It was developed at the Herlev University Hospital in Denmark in collaboration with Technical University of Denmark, that is DTU. So these are the some example images or some example cells of Neurope database. Neurope database, where the Neurope database is divided into mainly two types of cells, normal and abnormal, where the normal cells are subdivided into three types: superspecial squamous epithelial cell, intermediate squamous epithelial cells, columnar epithelial cells, and where the abnormal cells are subdivided into four types of cells, that is mild dysplasia, moderate dysplasia, severe dysplasia, and carcinoma in situ. So carcinoma in situ is the most invasive type of cancerous cell. So here you can find one superspecial squamous epithelial cell where the shape of the cell is polygonal, or you can see here the cell is also flat and the size of the cell is the largest among all other squamous cells, where the cytoplasm area is transparent and pink and it is abundant. That means it has enough amount of cytoplasm area, and the nucleus is also small and it is centrally aligned here. Okay, so next in the figure 10 we have intermediate squamous epithelial cell. Here the shape of the cell is polygonal or slightly oval, that is size of the cell is medium superficial squamous smaller than other squamous cells, and the cytoplasm area is moderate and the nucleus is larger than squamous cell. Next, in the figure 11, here we have columnar epithelial cell. So the shape of the cell is columnar, shape of the cell is variable, that means moderate, and the nucleus is elongated. So we are in this slide, that is figure number 11, columnar epithelial cell. So the shape of the cell is columnar in this type of cell, and the size of the cell is variable where the size is not fixed, and the cytoplasm area is also moderate. Nucleus is elongated, that is long and thin nucleus will be shown in this columnar type of epithelial cell. Next, figure 12 shows the mild dysplasia cell where the shape of the cell is irregular. The size of the cell is also variable. It is smaller than other dysplasia cells, but it is larger than the normal one. And cytoplasm area is also moderate but less abundant. That is not enough to discriminate. Okay. So here the nucleus is enlarged from the normal cell. In the figure 13, we have moderate dysplasia cell where the shape of the cell is also irregular and the size of the cell is smaller than the mild dysplasia, and the cytoplasm is reduced, and you can see here the cytoplasm is also denser than the previous one, or the normal cells. And the nucleus is also nucleus has also the irregular contour. That means the age of the nucleus is also not regular in shape. Then figure 14 shows severe dysplasia cell where the shape of the cell is round, it could be oval or irregular. The size of the cell is variable here, and the cytoplasm is also dense and indistinct, so that it could not be, we can't find out how many a region is in the cytoplasmic area is there. So next it is figure 15, that is carcinoma in situ cell. This one is the most invasive type of cancerous cell where the shape of the cell is remarkably irregular and the size of the cell is small. The total size of the cell is small, but the nucleus is the largest. It is the most enlarged nucleus area where it has dark in its feature is dark with abnormal mitosis. That means the cell is not divided into normally. These are the data statistics of Nurup database. So in Nurup database, in this Nurup database, 74 images are superficial squamous epithelial, that is the normal cell, and 70 images are intermediate squamous epithelial cell. This is also normal type of image, and then 98 images are columnar epithelial. So in total, there are 242 normal images, that is superspecial, intermediate squamous, and the columnar epithelial. And the in abnormal case here, we in the Nurup database has 182 images of mild dysplasia. So mild dysplasia means the lesion is low grade lesion, and then 146 images are moderate dysplasia, and then 197 images are severe dysplasia, and 150 images are squamous cell carcinoma in situ. That is, in total, there are 675 abnormal images. So total number of pap smear images, that is normal plus abnormal, there are 917 images in the Nurup database. But to the best of my knowledge, there is no existing cervical cancer database of the Northeast Indian population. So here it is AGMC-TU pap smear cytological image database. The AGMC-TU pap smear cytological image dataset annotated with ground truth images was prepared by the team of Biomedical Infrared Image Processing Laboratory of Computer Science and Engineering Department, Tripura University, in collaboration with Pathology Department of Agartala Government Medical College, AGMC, and Govinda Vallabh Pant, GBP Hospital, Government of Tripura. So these are some example cell images of cytological images of AGMC TU pap smear cytological image dataset. These are the statistics of this database. So in our database is actually composed of 100 conventional pap stained cervical cell images. The images are taken randomly from 20 patients, that is 5 images per patient of the Northeast Indian region population, and found defeating for image analysis, acquired through a digital camera adapted to a light microscope. Images of this database are stored in JPEG format of 132 by 158 pixels. Among 20 patients, 10 patients are found normal and 10 patients are found abnormal under the observation of medical experts of pathology department of AGMC and GBP Hospital, Government of Tripura. These are the major contributive features of this database. The dataset contains newly created standardized pap smear images focusing on cervical cancer screening. Creation of such a dataset will expand the area of research in automated disease diagnosis. The dataset contains ground truth images of suspicious nuclear region in the form of binary marks. The ground truth marking is performed under the supervision of radiologist. Pap smear images of normal and abnormal subjects are present in the dataset. Each pap smear image of the dataset is labeled by consulting with the medical expert in this domain. Data acquisition protocol. PEP test or Papa Nicolau test is a procedure which helps to remove a small sample of the cell from the cervix. After getting the sample cells, they are looked at under a microscope to see if they are normal or abnormal. To identify abnormality and to collect valuable information for medical research work, it is necessary to maintain a standard and universal acquisition protocol during the PEP test acquisition. So the first step is patient selection. The second step is pre-analytical patient preparation. The third step is PEP test methodology, cell staining, then capturing samples using light microscope. By following this data acquisition protocol, the team of Biomedical Infrared Image Processing Laboratory of Tripura University and AGMC Govinda Vallabh Pant Hospital has created this AGMC-TU pap smear cytological image dataset. Step 1, that is patient selections. The patients came to whoever came to the pathology department of AGMC and GBP Hospital, Government of Tripura, from different places of Tripura to screen for cervical cancer or to detect precancerous cell or check for abnormalities or infections like human papilloma virus HPV under the medical expert has been selected for the data collection. The next step is pre-analytical patient preparation. To achieve the best possible result, we have done the PEP test on the patient by taking care of the necessary steps. So patients should avoid having a PEP test during the menstruation cycle because the additional cells can obscure the cervical cells. To get the best result, the test must be done in the middle of the patient cycles, that is 10 to 20 days after the first day of the patient's menstrual period. The patient should avoid the sexual intercourse 48 hours before the test. The patient should not touch the 48 hours before the test. The patient should not use vaginal creams, foams, flames, or jellies such as medications inserted into the vagina for 48 hours before the test because these products can wash away or hide abnormal cells. The patient should avoid having the test during the treatment for any cervical or vaginal infection, and she should wait for some time until the treatment has ended. The made patient's bladder empty right before the pap test. A full bladder may cause having the test uncomfortable. Step 3, that is the pap test methodology. To do the pap test, the medical experts gently place a speculum into the vagina of the patient. A speculum is a clear plastic or metal device. It separates the wall of the vagina so the doctor can see the upper part of the vagina and cervix. The medical expert uses a small stick or spatula to gently scrape the surface of the cervix to pick up cells. In some cases, a special brush, also called cytobrush or cytobroom, is used to collect cells from the inner part of the cervix, which leads into the uterus. Samples of the tissues from the vagina can also take during the pap test. Here in this picture, we have in picture figure 18a shows the speculum. Here it is figure B is the cytobome, and figure C is the cytobome with the slide, how the smear is placed into the slide. Cell staining. So after collecting the cells, the doctor or nurse smears them into a glass slide or places them in a container filled with a special liquid, which is also called a liquid-based pap test. In our study, Papa Nicolau stains were used for identification of features related to nucleus. The figure depicts the steps used for cell preparation. Papa Nicolau stain needs intermediate rapid fixation in alcohol to preserve the details of the cells and to avoid the smears dying out. Ethanol 95% were used for fixation. Here in the figure 20 shows how to spray ethanol 95 into the glass slide. These are the objectives of Papa Nicolau staining. The objective one is to define the cell nuclear to aid in the identification of nuclear abnormalities of cancer cells. Objective 2 is to stain the cytoplasm and make it transparent for visualization. And the objective 3 is to differentiate and identify certain cell types such as acidophils and basophils. Papa Nicolau stain includes both acidic and basic dyes. Acidic dye stains the basic components of the cell, whereas the basic dye stain the acidic components of the cell. So these are the stain we used in our case. So hematoxylin stain, which is a natural dye, it stains the nucleus part of a cell. This dye binds to sulfate groups on DNA because it has a high affinity for nuclear chromatin. Next, it is orange green 6 were used. It is an acidic counter stain that stains the cytoplasm of mature keratinized cells. Components of white are stained orange at different intensities. Polychromic stain, which is a mixture of light green SF and eosin G, were used as a cytoplasmic stain. So figure 12 shows how the cells looks after using hematoxin stain, and the figure B shows how the cells looks after using orange green 6 stain, and then figure C shows how the cells look after using polychromic stain. So clearing in xylol produced cellular transparency prior to mounting. So mounting was after clearing, the mounting was performed with DPX, which is a mixture of glycerin, plasticizer, and xylene. The next step, that is step 5, is capturing samples using light microscope. After staining process, the samples are placed under the light microscope. A light microscope is an instrument that uses visible light and magnifying lenses to examine small objects which are not visible to the naked eye or in finer details than the naked eye allows. So the minimum, equipment required for the microscope, three, huh? Am I audible?
Yes, yes, audible Shobhishta. Proceed.
So, in AGMC-TU cytological image database, the sample images are taken through a digital camera Olympus SP350 adapted to a light microscope Olympus 3 into 41 having 80 megapixel resolutions, and the images are stored in JPEG format of 132 by 158 pixels. Next, ground truth generation. So, ground truth generation is a crucial work to verify the competence of a framework. Generally, ground truths are generated using manual segmentation by the medical experts to visually identify the presence of abnormality. In the terms of medical image processing, these ground truths can also be used to measure the performance of different segmentation algorithms. Nurup et al. And Martin et al. Considered CHAMP software to segment the pap smear images. But unfortunately, for our case, CHAMP software cannot provide a satisfying segmentation performance, especially for the abnormal ones. GNU image manipulation program tool, that is GIMP, has been used for generation of the segmented nucleus region to segmented our nucleus region. GIMP is a cross-platform image editor available for Linux, Mac OS, Windows, and more other operating systems. It is a free software. One can change its source code and distribute changes. GIMP for simple graphics need without having to learn advanced image manipulation methods. The medical expert performs segmentation using this software for generating the ground truth images. So this is the GUI, that is the graphical user interface of GIMP software. Here we have the link for GIMP software. You can go to this link and download it and install into your system. And these are the steps written in the left side, that how you can uh, create a ground truth of a cell image.
Acha Shobhishta,
Yes, sir.
Shobhishta, am I
Yes, sir.
audible? Acha,
Yes, sir.
one thing I have to convey to all the candidates, all the participants, that uh, this is a annotation of ground truth. Are you
Yes.
know? I am asking to the participant. Are you know the what is ground truth? What is annotation? Acha, annotation is the occupancy or the region of interest, where that is which is marked by the physician or the that is the doctors whenever the region of interest is uh, their occupancy of the cancerous cell or the nucleus part of the cytoplasm part is uh, occupy the part is region of interest is that is uh, indicated by the doctors. For the computer, this is most important. Without any ground truth, supervised learning is not possible in machine learning. So, this is most important for the supervised learning. So, Mr. Proceed now. Thank you.
Okay, sir. Thank you, sir. So these are the steps to generate ground truth images. So at first you have to go to this link and then download it. So after that after the installing, you will see this type of GUI on your screen. Then you have to go to this file tab, and then from this file tab you click on here open. Then you select the folder from where you want to open the image file. Now here you can see the image file is open in the GUI. So now you can do your work. So from the free select tool, you select this tool, free select tool, and then you select the region of interest. For our case, the new world neurology region. So by clicking on invert, the your selection will be inverted. Now the whole section other than this nucleus area will be selected. Then you again to go to this edit tab, and then from this edit tab you select this fill with FG color, that is black. Now you see the region, where you want to save it, and you just rename the name of the ground truth image, and then you click on here export. So in this way you can create or you can segment the nucleus of the nucleus area of the cell. So this is how we have created uh, ground truth images of AGMC-TU pap smear cytological image dataset. These are the reference. Now I will show you how this GIM software works. Now this software is opening. Okay. So at first from the file tab, you have to open the image that for what you want to create the ground truth image. So this is the image. Now you select this free select tool, it is here. In this way, you can select the edge of the image nucleus. That means we want to fill this region of interest into white. Now you go to select tab and just click on invert. It means you are inverting your selection. Now you can see here the area other than this nucleus area is selected. Now you go again into edit tab and click on fill with FG color black. Now you see this is the ground truth image of our cell image. But we have to save it now. To save it, we have to go to export as. And here you choose the folder from to where you want to save it. Already created. So this one is our round tooth image. Thank you.